said, this is one of the flagship projects, data science for personalized health. So we have quite a few PIs on the team. My co-PI is Mike Snyder from the genetics department. And then we have Ivo Oslu, Zaki Wiseman, Dan Bonnet, Andre Montanari, Moshan Bayati, and Ewan Ashley on our team. So today, a lot of the PIs are actually here, except for the medical school folks. So there's a very big meeting on the East Coast, annual meeting on genetics. So they're gone. So now you get the geeks. So basically today you get the geeks. The medical school people have gone. So what I would do is I would spend 20-25 minutes to have an overview of our project and updates. And then I will have the other co-PIs to come up as well. And then we can engage in some discussion. Cool. All right, so the impetus of this project is the observation that in recent years, there's been a huge omics data revolution. So omics refers to, so the starting point was genomics, but now it's spread to all kinds of things that you can measure about the individual. So as we all know, high throughput sequencing has a 7 orders of magnitude decrease in cost. So that was one of the big impetus of gathering all kinds of data about the molecules in our body. At the same time, we have an accelerated adoption of EHR, electronic health record, as well as smart sensors collecting health data. Okay? So this project is really about how to integrate all this information that we collect and do something useful with it. Okay? In particular, our focus is around, so this is about data science. So first I should talk about the data. So the data we're focusing on is an ongoing project in the medical school headed by Mike Snyder on doing omics profiling of 100 pre-diabetic patients. So with these 100 patients, we're collecting a lot of different omics data, okay, all these omics data. Genomics, transcriptomics, proteomics, etc., which I'll go into a little bit more detail later on when I talk about the projects, as well as sensor data and medical test results. So we want to combine all that onto creating a personal omics profile and then do something interesting with it. Okay? So I'll spend a few slides just to give you an idea of what the data collection process is like, because after all, if you want to do data science, you need to understand how the data is collected. So this project is currently 3 years, ongoing in the second of the 3 years. Basically, for each patient, data is collected at different time points. So every 3 months, the participant comes in to do some blood tests, etc. They are also undergoing, so during this time, they may have, say, infection, viral infection, for example, some illnesses, and they ask them to come in when this happens, and we do more tests, more frequent tests when they're sick. When they undergo some stress, we also apparently ask them to come in, as though they're not stressful enough, to come in and do even more tests. So this is quite a big requirement on these participants. So if after this talk you're kind of interested in joining this cohort, come and talk to Tejas, who's our representative here from Mike Snyder Group. More interestingly, they are also subject to some forced diet change to overfeed them and to see how they react. So this is not only a passive observation, but active as well. I wish Mike Snyder were here, because then he can tell you a lot of interesting stories. Okay, so basically longitudinal studies, longitudinal tracking of 100 people. So for each of these time points, what is the data being collected? Basically, there's about 14 omics assays, okay, being collected from blood samples, all these kind of different biological molecules, as well as stool and urine, and sometimes also some nasal and skin samples. Okay, so this is one time point. Now, in addition to these every 3 months measurement and also the measurement during the viral infection time, there's also continuous monitoring through sensors, through these glucose monitoring, smartwatch, ECG heart monitoring, etc. Okay, so this is the data being collected And just to give you a sense of how much data is collected right now, roughly at this point, across these 100 participants, some are earlier in the project, some are a bit later joining the project, so you have different number of time points for different individual. Average around, say, 20 time points on each individual. Okay, so 850 time points collected. Okay, actually not 20, 30, 40, I guess. Okay, so a total of 131,350 essays already run, and it's an ongoing process. So in fact, a lot of this work on this project is really how to integrate all this data together. And Tejas is very involved in that process together with a few people in Mike's group. Okay, so these are some examples of essays done. Okay, so, all right. So basically what's happening here is that this kind of data is being collected as we speak. Okay, this is a data science project, so we want to do some interesting data science on this data. Okay, so about a year ago, when SDSI asked for a proposal, we thought this is a really cool data set to be able to do some interesting data science. So our goal is to try to bring some data science tools, either existing ones or adapt existing ones or invent new ones to put to bear on this type of data set, this omics data set, via interaction between data scientists and domain experts. So you can see on our team, there are two people from the medical school, Mike Snyder and Ewan Ashley, who are domain experts, together with their group members. And then there are a few other data scientists from statistics, electrical engineering, computer science, and the business school. Okay? So basically, you can break up this project into 3 parts. So there are 3 key stages about data. First, we want to have devices to measure and process the data. And here the data science question is, well, how often should you sample? As you can see, the samples are kind of non-uniform. How do we deal with that? Imputation, for example, there are a lot of missing data. So you ask the participant to come in every 3 months, but sometimes they get busy and they forgot to come in. After all, you're not really paying them too much, so imputation. So how do you fill in this missing data? So these are the problems here. Now that you collected data, you need to manage the data. Okay? So 2 particular problems we looked at in the managed data front. One is compression. There's a huge amount of data being collected. I'll give you some idea of the scale of the data later on. How do we store it? And how do we store it in an efficient way so that we can utilize it? Okay? So these are the questions we asked. Privacy and security. So a lot of these data, we scaled out this project. When there are 100 participants, the issue of privacy is less because these 100 people are very motivated to help out. But if you want to scale this to the general wide population, then there are a lot of privacy issues. So privacy about sharing data, and that is one problem we are also looking at. And at the end of the day, you get this data, and you want to utilize the data to do prediction. So predictive analytics is sort of another aspect of the project we focus on. Okay. All right, so how do we organize this project? So first, we have domain experts, and we have data scientists. So we form several collaborative subgroups. Each some of them involve a data scientist and a domain expert, and they collaborate. Okay. Second, we meet as a group once a month. Okay. So since the beginning, which is about 7, 8 months ago, when we heard that this project was funded, we developed 4 ongoing projects involving various PIs. And I will briefly, so the rest of the talk, I will briefly talk about these 4 projects. The level of details, because of time limitation, will do no justice at all to any of these projects. So what we do is 2 things. One is we'll have the PIs will all come up after this talk, and then you can ask them any question. Don't ask me, of course. Second, 3 of these 4 projects will have a poster in the afternoon. The poster session, I believe, starts at about 4:15 or so. And there, you will get to talk to the students and the postdoc, who's actually, of course, doing the actual work. They can tell you more about the details. Particularly, I will not spend that much time on the methods, but I will show you some results. Okay? initial results. All right. So 4 projects. First one is a downstream friendly omics compression. As I mentioned, compression is an important problem. Okay. So the type of data that we need to compress, a lot of them, is basically from this high throughput sequencing. So from high throughput sequencing, you generate a huge amount of data. So each measurement could be almost a few gigs of data. Okay. So in this data, the question is, well, how do you compress it? Okay. So the first observation that was made is that actually, although you are interested in biological information, most of this data is actually not directly about the biological information, but about the quality of the information. It's so-called quality values. Okay? So if you are interested in compression, this part, which is the biological information, is kind of only a small piece of the story. The biggest gain in compression is actually this blue part, which is the quality value. Okay? However, biologists and medical school people are very sensitive about compressing the information. Because if you start compressing them and losing information, then they get very unhappy because it costs them a lot of effort to measure this data in the first place. Okay? However, an interesting observation is made by Zaki Weissman group, which basically says that actually the quality scores themselves are not sacred. They themselves are actually noisy information. So if you compress them, okay, it seems that you're losing information, but actually what you're doing is something positive, which is denoising the data. So what the big discovery of this group is to notice that if you apply a good compression scheme to compress this data, you're actually at the same time denoising it, and therefore actually improves the downstream processing. So one particular downstream processing that was analyzed in this work is so-called variant calling. So after you sequence the genome, the first thing you're interested in, one of the main things you're interested in, is what are the variants of this individual compared to the general population. Okay? Because the variants will tell you some risk factor to certain diseases. Okay? So the experiment here is to compare a standard pipeline with what happens after you do this compression algorithms that was invented by Zaki's group. Okay? And compare the quality, not in terms of how accurate you are in terms of recovering quality value, but how accurate, what it matters is how accurate the calling of the SNPs are. Okay? All right, so here's some results. Okay, so this is on chromosome 11 of one of the reference individuals that is deeply sequenced, 30X in this case, in this particular experiment. Okay, so here is an interesting point. So what am I plotting here? I'm plotting for each point, okay, the false positive and the true positive in the calling of the SNPs, okay? And this point is interesting because you can compress, okay, by 1.67 factor. So you save over uncompressed gain. But at the same time, you can actually do better in true positive and do better in terms of false positive. So fewer false positive, okay? And if you compress further, so this is a 10% compression. You're reducing it by 1/10 of the size, okay? You are still calling more true positive, but now a little bit more false positive. So here you can trade off now the performance with the compression. But this point is interesting because here you're saying that you can win in all coordinates. You can do better and compress, okay? So the natural question from this work is that, hey, you can do this compression and calling separately, and you can still gain. The question is if you jointly design the compression and the calling scheme, where you can still get further gain. And this is an ongoing work from Zaki's group. Okay? All right, so the first project is compression. This is the managed data. The second project is a privacy preserving. Okay? And this is a project between Don Bonnet and his student, David Wu. This is also in the managed data side. Okay? So as I mentioned, omics data is sensitive and private. Okay? However, very often, we need to analyze this data. So imagine we're collecting this data over, say, a few million people. We need to estimate some overall aggregate statistics across these individuals. But each hospital may not be very interested in sharing the data completely in the open. So this project is to say that, well, how can you collect the joint statistics from each of these individual, say, hospital, but at the same time be able to get the information you want, for example, the statistics, and yet not reveal information to the cloud. So they participate in this competition called I-competition, okay? And here's a task, okay? What is the task here? So here we have 2 hospitals. They want to encode the genomics data, the omics data, okay, to the cloud. So this is outsourcing the genomic analysis to the cloud. The goal is to design an encryption scheme so that the cloud can do the job, in other words, collect the statistics that they're interested in, but at the same time learn nothing about the genomics data other than the answer they compute. So they apply this general framework to 2 particular problems. One is estimating minor allele frequency. Okay, so how rare these alleles are, which is an important parameter in a lot of genomic analysis. And David Wu got first place in terms of runtime in this task, in this competition. Task number 2, which is also important in genomic analysis, which is to compute the distance between 2 genome sequences. So now this task is that can you submit 2 individual sequences to the cloud computes the edit distance returned to the sender, the clients. At the same time, the cloud does not learn anything other than the edit distance between these 2 sequences. Sounds like black magic. And I'm happy to defer all questions to Dan Bonnet after this talk. Okay. All right. And there's some future work on trying to understand how do you do case and control studies, basically. In other words, finding genes that are responsible for certain diseases using the same idea, distributedly. Okay? All right. The third project, now we move on to the predictive analytics part. So now we want to ask questions about, okay, so you collect all this data. Okay, so what can you do interesting? What data science problems can you do interesting on this data? Okay, so this is a joint work between my group and Mike Snyder group. Okay, biological pathway identification and disease prediction. Okay, so just to bring back the general setting of the problem, this is a time point of one individual, one of the 100 individuals. It just happens to be Mike Snyder. This is complete coincidence. Okay, but we like him. So we spent a lot of time on this data. We really like it. So different time points corresponds to healthy state, okay, or infection state. Here we focus on viral infection. So I give you a time point. All right. Can you tell me whether this time point is when the individual is healthy or when the individual is in a viral disease? Okay. That's the problem we're after here. That's the prediction. That's a prediction problem. Okay, so we start working this problem in the past few months. So the first observation is that the data is high dimensional, okay? If you combine all this data that we said, the genome, the transcriptome, which is the RNA, the proteome, which is the proteins, and the metabolome, which is the measure of the metabolites, if we combine all this data and try to do prediction, let's look at the dimension of the problem. First, there are about 3 or 4 million SNPs. SNPs are these variations on the genome. 20K to 100K transcripts, these are RNA sequences. 6 to 8K different types of proteins. We have measurements on the concentration of each one of them. And 8 to 10K of metabolites, okay? So you put all these things together and just run some, say, linear regression or some significance test on this huge dimensional vector. You don't get something very interesting. Because what happens is that you get a lot of false positives, okay, false discoveries. So we kind of worry about that. So our whole problem was really trying to understand how to integrate this problem, how to integrate all this data together, all right? So again, we're interested in the prediction problem at the end of this project. However, the first problem we looked at is, as anyone knows in machine learning, a key step in machine learning is really to extract the relevant features from the data. So the problem that we've been working on the past few months perfect, is how to extract the relevant features from this huge dimensional data set. And then the second step is to use these features to do predictions. So we haven't done the second step yet, but I'll report some results on the first step. So we need a framework to integrate all this data. And luckily, we have Tejas, the biologist on the team here. And then we say, hey, why don't we use this concept called a biological pathway, which allows us to, as a hyperfeature, to integrate this data. So instead of looking at gene one at a time, or protein molecule one at a time, biologists knows that everything is connected together through pathway. Okay? So there are databases, okay, which is community curated, which is biological, meaningful, structured collection of features. And there are about a few thousand of these pathways. Okay? Each pathway is a connection of a bunch of proteins and metabolites. Okay? So we said instead of learning the features on a very low level, like the genes, why don't we try to learn which pathways are relevant for this particular disease, say viral infection? Okay? So we developed some new method to solve this problem using Gaussian graphical models. The details will be after, and I'm running out of time a little bit here. So we run a significance test, and we find a few interesting pathways suggested by the significance test. Okay? Well, okay, one of the main problems that we face in the data science is how do we know we're doing well? What is the ground truth? Unfortunately, we're talking about science here, so we don't know the ground truth. However, what we do know is that there are biological literature out there which tells you which path. We have already some background on the pathways that are relevant for certain diseases. So there's some existing knowledge, and we can use that to validate whether we are on the right track here. And so what we find is that the pathway we discovered, we selected using this method, actually encompasses a wide range of metabolic activity associated with viral infection. Now, I would love to explain to you all these different diseases. But first, I have no time. Second, I have no idea what they are, actually. Okay? I must be honest with you. But you can certainly ask Tejas about it. But one interesting thing is that this one called nonsense medicated decay is actually something which is discovered related to viral infection only in 2014 in the publication. Okay? And we have also done some comparison with just by doing a standard gene-by-gene analysis. Okay? And we find that we are discovering a broader range of pathways. So details in the afternoon. Okay, so we want to incorporate our data. And one very interesting problem we were thinking about, okay, do you think I can ask for a 3-minute extension on my measly 20 minutes? Okay. I think I need 3 more minutes. Is it okay?
No, that's okay.
All right, thank you very much. One very interesting problem is the following. So far we're focused on one individual, Mike Snyder. He has the most comprehensive data set. But we have other patients, other subjects, having more and more data collected. So the next stage of the process is now, a project is now how do we look at all the data of many individuals? And we turn out that there's a problem here, is that different individuals, the data actually looks quite different because of the individual aspect. So our problem is the following. Each individual only has like 20 or 30 time points. So it's more data. So the number of time points is small, but each time point is very high dimensional. Question is, how do we use the fact that we have 100 participants, and therefore a total of a few thousand time points, and use that to enlarge the statistics, to be able to get accurate statistics, while being able to do prediction on individuals, each with their specificities. That's the problem we've been racking our brain to try to solve. And our idea is to use the genome, because different individuals have a different genome. In some sense, the genome identifies the individual. And we want to use that to, we have some ideas, but not really very concrete at this point. Okay, the last project I want to talk about is again on predictive analytics, but now a little bit high level. Okay? So this is work with Motion by IT, Andre Montanari with a student, Sonia Basker. Okay, so here's the problem. Right now in hospitals, they run huge number of labs. The huge number of possible labs that you can run to try to predict certain diseases. So the standard workflow is that each disease, you try to figure out which labs is good predictive for that disease. But if you do that separately, then you end up having to run many labs because there are different diseases may require different labs. The key observation of this project is that you can save cost by actually trying to find a common set of labs that simultaneously predict well for each of the many diseases. So in this project, they looked at 9 different diseases. So the problem is how do you save medical costs? So we haven't talked about cost too much, but cost is certainly an important issue on reducing the data and at the same time maintaining the prediction accuracy. Okay. So if you look at existing methods like logistic regression, okay, then if you look at the cost accuracy plot, then you'll find high cost, and low accuracy. So here, logistic regression will give you many, many tests to run, basically. Now, if you do some kind of lasso, sparsity, and say that you only want a small number of labs, then you decrease cost and accuracy actually is better. What they've done is instead of doing task-by-task learning, single task learning, one for each disease, they do multi-task learning, which is to try to find a common set of labs which predict well for all the diseases simultaneously. And they show that the accuracy can be maintained while the cost is significantly reduced because now you don't have redundant labs that you need to do for different diseases. Okay? So we're hoping to bring that kind of expertise into this project by combining with other types of data like sensor data for the coming year. Cool. So I see that Hector's already here, and I'm finished.
Nice impression. Good. Thank you very much. We still have a few minutes for questions for David or his collaborators.
Yeah, so I'm thinking of bringing my collaborators up here. Are we going to have, is that good? Because they're scattered all around the room.
Yes, there's Dan and Tucky and good, yeah. So I have one question to sort of get you the juices flowing. Oh, thank you. Did I miss something, or you didn't mention sort of the traditional records? I mean, when you go and see the doctor, they make notes and they you have medicines given to you and so on. So is that part of the data set?
Yes, in fact, it was very quickly. There was one slide when we talk about omics assays.
Right.
So omics assays are the blood, you draw blood from the patient, and then you collect, you analyze, and you get these omics data. But at the same time, there are also medical tests as well.
Okay.
Yeah.
So that's all.
So it is, after all, a project in a medical school. So they do have doctors there and so forth.
Other questions? Yes. Please.
Let's use the microphone so we get it on the recording.
Okay.
Was it you, Chris? No.
I thought it was very interesting in your prediction problem that you were starting to see benefits of using the biological pathways as built into the model.
Yes.
So can you say a little bit more about prospects for learning new pathways from the huge amount of data you're working with? So as well as the ones that are in the literature already, is there a potential there?
Yeah, so that's a good question. Honestly, we have not. So this project has been going on for a few months. So we first wanted to test whether actually we can find some interesting pathway within the database. But you're correct in the sense that the database is community curated. So there are potential for discovering new pathways. Yeah, I don't have a very concrete answer to that question, but it's a good question. Yes, Tejas.
So there's basically like 2 parts to answer this question. One, I think as David mentioned, is that the pathways themselves are defined by what the community curates. So we are limited in a sense by the information that's out there. The potential for discovery is when it comes to, so the nonsense-mediated decay pathway that you mentioned, it's a normal pathway that functions in normal cells. It's only recently been reported that human cells use it to fight against viruses, and that was reported as recently as 2014. If I'd have seen that pathway, if that paper had not been there, I would have probably thought, oh, well, this is probably something it's probably a junk result that came up. But we would think about, well, is this something, is this in some way involved in viral infection? Are there ways that this pathway could act to mediate defense against viruses? So there are, in that sense, yes, we can discover novel pathways that have not been seen before, and we can come up with sort of mechanistic answers to figure out how these pathways mediate viral disease. And in that sense, we can learn new biology. So that's basically, yeah. That's it. Sorry.
You want to give a shot at the microphone?
This is a fairly boring question, Dan, but for you, on the way to the cloud, you in and out, to, from, you encrypt and decrypt, but then the computation itself, is that cannot be done in the cloud, right? Because, and so how would that happen?
Yeah, it's actually the magic of distributed computation, and there are actually fancy encryption schemes that actually do allow you to compute data, to compute on data even though it's encrypted. And this competition that we participated in was exactly doing certain tasks on encrypted genomic data, like computing edit distances and chi-square distributions, even though the data is encrypted. So the computation actually is taking place in the cloud. The cloud gets encrypted, an encrypted result, sends it back to the hospitals. The hospitals can decrypt using their secret keys and get the results, but the cloud learns nothing at all. And the hospitals only get the data that they want. They learn nothing about their peer institutions' data. So generally, I should say this is like a very fruitful area of research. This goes for all of you here in the audience. If it's just a matter of just protecting the data by encrypting it, that's not an interesting research problem. That's something that we know how to do in our sleep. If it's a matter of actually you have data that's stored on distributed systems, for example, 2 different hospitals, they want to compute on the union of their data, but they don't want to reveal the data to one another for regulatory reasons. Those are exactly the kind of problems we love to think about. And as David mentioned, actually, we're starting to work now with Gil Balgirano on identifying defective genes. So you have like a population of 100 people, they all show the same defect, they also show the same symptoms, and you want to understand which gene is causing those, this genetic defect. So they can do some sort of a computation that would reveal what the defective gene is without actually revealing anything else about their genomes. So those are kind of beautiful privacy requiring problems. We love to work on those. If you have more examples of problems like that, essentially, again, you have data that you would like to process it as centrally, but you can't centralize it for regulatory reasons. Those are problems that we would love to help you work on.
Good microphone. You can use this one here. Question?
I'd like to follow up a little bit on that one. In this area of privacy-preserving computations, there are a huge number of interesting techniques that are emerging. Semi-homomorphic encryption, differential privacy, software guard extensions. Do you see sort of an evolving big picture about when and how to use those techniques, strengths and weaknesses? How are you organizing your research to help provide insight and guidance to people?
I think that's a really good question. So let's see. So I'm interested in kind of the cryptographic techniques that can come into play here. So exactly things like distributed computation. Obviously, if you care about the privacy of the results, you can actually throw in differential privacy into the distributed computation. I think what Intel is going, the direction that Intel is going is a little different, in that what Intel is providing with SGX is kind of a hardware platform where trusted computations can take place and no one can peek into the hardware. So that's a fantastic area too, and as you know, we now have an Intel SGX machine, and we just got it like literally last week. So we're going to start playing with that for the purpose of distributed computations as well. The difference there is, with the crypto applications, of course, the cloud learns nothing, period. There's nothing the cloud can do. With hardware-based solutions, things are more efficient, of course, but security is only as secure as the hardware. So those are kind of the fundamental differences, and we're kind of exploring both directions. Yeah.
Okay, we have time for 1 or maybe 2 more questions. So one here.
Something about compression, maybe.
There's one other person here. Yeah, read next.
I was very intrigued by the last project that you were presenting. I was thinking about the fact that most of the time these machine learning tests oftentimes give you a probability of that maybe you have this risk factor for this disease, let's say 0.8 probability or something like that. But then the confidence interval is like plus minus 0.5. And oftentimes in these computations, we don't really carry these confidence forward. And when you go to the multi-task learning, as you were calling it, I would imagine that this problem would be even more severe. I was just wondering if you wanted to comment on that.
Great question. So we can calculate uncertainty around the prediction as well as uncertainty around the features that are selected for the prediction. But our objective in this project has been so far calculating the expected value, which is a prediction itself, and we can see the area under the curve in terms of that metric. We don't lose anything, and what happens, we are not picking noisy features. All the noisy features will be dumped because of that we are learning multiple diseases at the same time. But it is possible to calculate. There are methods that in the literature that allow you to calculate the uncertainty around the prediction as well when you use these regularized methods.
Okay, good. And our last question?
Thank you. Rick Barron, Swiss Re. You mentioned the use of wearable devices. I was very interested to know if you did any analysis to understand the relative value of the data that you got from wearables compared to the data that you were getting from fluid testing.
So there's a wearable expert here.
Hi, I'm Jessie. I'm also a postdoc in Snyder Lab, and I'm working on the wearables aspect of this project. So the wearables is in the really early phase, so we're still working on getting the larger population of the wearables data. But the initial studies on our kind of N of 1 have shown that the wearables do actually predict some illnesses. So if, for example, it's an illness where we expect that the heart rate should go up, that's something that we can detect. So now the next question is to tie it all together. So work in progress.
Okay, good. Maybe you can. Okay, we're doing things the same. Okay, so I guess that completes this talk. Thank you very much, and thanks for bringing all your co-investigators. I think it was a great idea. Thank you.